In this video, we continue our discussion of the endocrine system. I'm your instructor, Megan Workman, and I'm excited to guide your discovery of two types of hormones, peptide and steroid hormones. This topic will make a lot more sense if we warm up our brains first. So let's perform the following scholastic jumping jacks. What do you remember about the structure and function of plasma membranes? Recall that membranes are comprised of phospholipids that zip together like this, with their hydrophobic fatty acid tails associating in the middle and their hydrophilic phosphate heads lining the inner and outer edges of the membrane, interacting with the aqueous solutions on either side. This structure, when zipped together, creates a thick layer of fatty acids in the middle, which are lipids and therefore hydrophobic. This structure creates a barrier, preventing mixing of the solutions on opposite sides of the membrane. Water and water-loving molecules cannot easily diffuse across, nor can large molecules like this peptide. But small lipids, because they are hydrophobic, can pass right on through. Next, let's remember what we learned in a previous lesson when we began thinking about the endocrine system. You previously learned that hormones are chemical messengers produced in an endocrine gland that travel throughout the body to their target cells, which is any cell that has a matching receptor. Keeping all of this in mind, you're well prepared for the next challenge. By the end of this video, the successful student will be able to relate the contrasting molecular properties of peptide and steroid hormones to differences in their production, storage, and release from the endocrine cell, transport in the blood, and receptor locations within target cells. The majority of hormones produced in our body fall into two categories, defined by their molecular structure. One group is made of amino acids. These are hydrophilic, so they cannot diffuse through membranes. The other group is made of small lipids, which are hydrophobic and therefore can diffuse through membranes. This difference in capacity to move through membranes predicts every additional contrast you should remember about these hormones. The amino acid-based group are called peptide hormones, while the lipid-based group are called steroid hormones. You may have heard of some of them. For example, insulin is a peptide hormone, and the sex hormones, like testosterone, are steroid hormones. Let's check in. Based on their contrasting capacity to move through membranes, which type do you predict more easily escapes the cell from which it's made and diffuses into its target cell? The answer is steroid hormones. Steroid hormones are produced by the cortex layer of the adrenal glands and by the gonads, the testes and ovaries. Cells in these glands are similar to any other eukaryotic cell you've studied. The organelle that synthesizes lipids, like steroid hormones, is the smooth endoplasmic reticulum, but some steroids are also produced by mitochondria. Zooming in on these organelles, we see that steroid hormones are released into the cytoplasm and are free to diffuse throughout the cell, including to the cell membrane. Since they are small lipids, they are not prohibited from exiting the cell and they easily diffuse right across the cell membrane. So, there is no mechanism to retain them or stockpile them for future use. Steroid hormones are secreted at a pace that matches their production. How about this? Which type of hormone can be contained by membranes, including within the membrane of a vesicle? Which type can be stored for a future release date? Peptide hormones. There are many different types of peptide hormones, and different glands produce, store, or secrete specific ones. Because they are polypeptides, you might remember which organelle synthesizes them. Ribosomes embedded in the rough endoplasmic reticulum. The peptides then travel through the endomembrane system to the Golgi apparatus, where they are packaged into membrane-bound vesicles, from which they cannot escape. These vesicles may accumulate, producing a stockpile of peptide hormones. Vesicles release their contents to the extracellular space via exocytosis, shown in this image. The pace of exocytosis, which is how peptide hormones are secreted, may or may not be regulated. A scenario in which exocytosis occurs at a relatively constant pace is called constitutive secretion, as shown in the upper example. Contrast that with the lower example in which peptide hormones have been stored in multiple vesicles, and exocytosis is mediated by that ligand-bound receptor located on the outside of the cell. This ligand is a chemical messenger that caused this target cell to secrete the little red peptide hormones. This scenario is an example of regulatory secretion. So peptide hormones are secreted via exocytosis, while steroid hormones simply diffuse into the extracellular fluid. From there, they both find their way to a nearby capillary and enter the bloodstream. Which type of hormone do you predict has an easier time circulating in the blood? The answer is the hydrophilic hormone, of course, the peptide hormone. Consider this interesting contrast. Peptide and steroid hormones experience opposite challenges because of their contrasting properties. Because peptide hormones are hydrophilic, they mix with water, therefore travel through blood easily, but they cannot diffuse across membranes, so they can't actually get inside their target cell. How on earth, then, are they supposed to deliver their message and influence the goings on within that target cell? Oppositely, steroid hormones, which are hydrophobic, diffuse right into their target cell if they can get there. But traveling is difficult for them because they are lipids and blood is water. So, literally, as lipids and water do not mix, steroid hormones don't mix with blood. So, how do they travel in the bloodstream? The body found solutions. First, I'll describe how each type of hormone travels in the blood. Peptide hormones are released by exocytosis into the extracellular fluid, which is never far from a capillary, into which the hormone diffuses, possibly through a fenestration or similar gap in the capillary wall. They are swept along by the current and exit all over the body, including near their target cell, where you see them binding here to a receptor that appears to be on the outside of the cell. Steroid hormones hitch a ride on a large hydrophilic protein like albumin that serves as a taxi. Most circulating steroid hormones are bound to such a protein, whereas most peptide hormones are free, meaning not bound to this type of taxi. When the steroid hormones detach, they leave the blood vessel into the extracellular compartment and may diffuse into nearby cells. Both types of hormones exit the bloodstream where they diffuse into the extracellular fluid, washing over potential target cells. The receptors for peptide hormones are located on the outside surface of the target cell's cell membrane, like the Y-shaped receptor in this image. This is called an extracellular receptor. Steroid receptors, on the other hand, are found inside the target cell, like these yellow bricks in the cytoplasm shown in the image. These are intracellular receptors. Now I recommend pausing the video to journal in your notes. Contrast the different challenges peptide and steroid hormones overcome during their journey from synthesis to receptor, and draw a cartoon target cell that has receptors for both peptide and steroid hormones. To review, peptide hormones and steroid hormones have contrasting molecular properties that predict all of their other contrasts. Peptide hormones are hydrophilic, while steroid hormones are hydrophobic. Because peptides can be contained within vesicles, they are storable, while steroids are not. This allows decoupling of secretion from production among peptide hormones, but not steroid hormones. In the blood, peptide hormones travel solo. They are free, not bound, while steroids are typically bound to their taxi. And when they reach their target cell, peptides cannot get in, so it's a good thing their receptors are extracellular. Steroids, on the other hand, enter their target cells without invitation and find their receptors within. Congratulations on all you've learned in this video. Do you feel that you have successfully achieved these learning targets? If I can clarify or address your concerns, please email me or pop into my virtual office hours by clicking on the stable link provided here. Happy studying!